Hi guys, this is activity 232, a lab all about biosecurity. I'm going to read our purpose statement as we prepare for this lab. Biosecurity is a growing concern in animal agriculture. Concentrated confinement operations that permit the transmission of pathogens make illness and disease, animal welfare, and economic concerns. Prevention and reduction of the spread of pathogens are crucial for today's agricultural operations. However, many common practices that farmers and ranchers use to raise animals also cause the spread of pathogens. Vehicles and machinery used to transport feed, supplies, medications, animals, products such as milk and eggs, manure, and people can carry pathogens between sites and facilities. The use of tools, supplies, and equipment at multiple animal production sites also spreads pathogens. The food industry uses a system of checks and balances called Hazard Analysis of Critical Control Points, also known as HACCP, to ensure food safety. In a HACCP plan, the industry identifies control points. Then they establish preventative measures for and regularly monitor these control points. Animal farm operations can apply the practice of preventing, reducing, and eliminating risks at control points as well. How can producers prevent the spread of pathogens? Several pathogens are bacteria. Scientists can grow bacteria in controlled environments to study specific characteristics. Three main characteristics assist in identifying bacteria, which are form, elevation, and margin. A bacteria's form is the shape of the colony or group of bacterial cells. Elevation refers to the height of the colony, and margin describes the edges of the bacteria. Can you use these characteristics to identify possible bacteria caused by not following biosecurity protocols? Let's get into the lab. We are going to be using Scenario 2 Farm Sanitation to perform this lab, which is a separate document on your assignment post called the Biosecurity Scenario. So what we have going on is we are a confinement swine operation, which means we grow pigs totally indoors, and we have hired a student to help around the farm. And we figure it's just fine that he wears his work boots in all locations between his own pigs that he has at his house because he's raising pigs for an FFA project, and also on our property with our pigs. So we're going to be using one of his dirty work boots to simulate this test. We're going to do 3 different samples to test out different biosecurity measures. And remember, we're trying to create a clean environment and keep everything good for our own pigs. We don't want to have bacteria grow in these. So I've got 3 Petri dishes labeled ahead of time for all of our samples. And you see there, I also have cotton swabs and then parafilm. We'll get to those in just a second. What we're going to be testing first off is just regular old shared farm equipment. We're not going to do anything to the boot at all. It's been from one farm to another. It's been from the backyard of this kid raising his pigs for FFA, and then he's also come to work on our farm. So it's got both properties mixed together. So you see there's some poop. FYI, this is my chicken boots, and it's just really just chicken poop. Anyways, so we're gonna swab the bottom of this boot, and this is sample 1. The technique you'll see me use for swabbing is I'm not gonna open up and expose that swab until I'm ready to actually do the swabbing right away. I'm then gonna swab the entire bottom of the boot, not just one particular location. And then what's hard to see as I do this is my motion. I am twirling and swirling so that entire tip of the cotton is being exposed. I'm not just using one edge, if you will. I'm then immediately going to swab that to my Petri dish. So I'm not letting it touch anything else. I'm not coming into contact with it. I'm going to immediately swab this Petri dish. This Petri dish has what we call agar or agar, depending on who you ask. It is a nutrient gel base, so that way if there is bacteria that I have just swabbed, it will feed it and it will start to grow so that we can see it after it's incubated for a few days. If the boot was clean and there was no bacteria, nothing would grow. All right, this is sample 2. We are going to do cleaned farm equipment. So I'm using a bucket of warm water and a scrub brush, and I'm trying to clean off all the debris from the grooves of the boot. There's no soap, there's no disinfectant. This is simply warm water and scrubbing. Once I've scrubbed the debris clean from the sole of the work boot, I'm gonna allow the boot to dry, and then I will swab. Now I'm going to complete our lab by doing sample 3, which is disinfected farm equipment. So I'm going to be placing this boot into a disinfecting solution. So this is the same bucket, I've cleaned it, but it now has warm water and a bleach solution. And I'm letting that sole of the boot be in contact with the solution for 5 minutes. I'm then going to come back, let it air dry, and then swab. Okay, so I have done all the swabbing, and I did 3 separate Petri dishes to do the full thing, because I think it'll be easier to show up on camera. But as you'll see in the worksheet, normally what you would do is just one and divide it into 3 separate sections. This gives us a better surface to look at though. So we don't know what is actually growing in here. There could be really nasty bacteria like E. coli that could spread. So what we use is parafilm, which is basically fancy saran wrap, to seal these Petri dishes. We're gonna seal around the lid to the dish so that we're not gonna open them again. When we do observations, we're simply gonna look through the dish, and then when we're done with the experiment, we'll dispose of the entire dish completely. That way, we're not posing a risk to anything. So all of these have been swabbed, and I'm gonna seal them all up with parafilm and then place them in the incubator to encourage bacteria to grow. So if there is bacteria present, we're gonna kind of speed up that process and be able to see it. And then in a couple of days, we're gonna come back and do observations. And if there is bacteria colonies growing, we're gonna try to identify what kind of bacteria they are based on the size, the texture, the color. There's all kinds of things with bacteria. If I could ever get this open, here we go. So I'm just gonna demonstrate for you. This is what parafilm looks like. It's kind of wax paper in texture, but it stretches like saran wrap. So that's what we're going for.